Good morning, children. Today we start with our practical sessions for class 11 as well as for class 12. And we are going to start with the food test, analytical experiment for carbohydrates, proteins and fats. I'm sure you all have carbohydrates, proteins and fats in your food every day. And you also need to know which food has carbohydrates, which food has proteins, and which food has fats in your daily diet. We are going to experiment on monosaccharides, disaccharides, proteins, as well as fats. We are going to take food items such as rice or wheat or gram seeds or milk. You can take anything. You can also make a powder of wheat and rice for carbohydrates. You can take glucose, glucon D for glucose. You can take any kind of seeds, mustard seeds, or fats. For fats, you can take, any kind of butter or oil. For proteins, you can take gram seeds. You can make a paste out of it, and you can dilute it with water, and then you can find out what kind of food is there, what kind component of food is there. Actually, when we do our experiments on glucose, children, when you do your experiments on glucose, you will find there's a color change. You experiment with Fehling solution A and B or with Benedict solution and then heat it. You will find there is a yellowish orangish precipitate. But at the same time, when you do this experiment with sucrose, which is a disaccharide, or any kind of disaccharide, you will not get this result directly. You have to change the disaccharide into monosaccharide first, and then you will do the experiment and you will get the same result. So when we take any kind of monosaccharide, whether we are taking some kind of juice, either we can do an experiment with Fehling solution A and B, adding equal amounts, or we can also take Benedict solution, and then when we heat it on the spirit lamp, we get a yellowish brown precipitate. This is definitely because they are reducing sugars. They change cuprous to cupric ions, therefore the color change. But at the same time, when I talked about disaccharides, they are not reducing sugars. They are not non-reducing sugars. So therefore, they will not change cuprous to cupric. So we need to change them into monosaccharides and then do the experiment. Proteins. When we talk about proteins, the smallest particle of protein or the smallest group of protein is called amino acid. And they have nitrogen in them. So when we do this experiment, either Millen's reagent or Xanthoprotic test or whatever, then this color change again to maybe a yellowish color, this color change is due to nitration. And this nitration will give the yellow precipitate. But at the same time, when we do with fats, just rub it on paper, you will find translucency, and that is a test for fats. You don't need to do any more tests on fats. That's all. We start with our experiments on glucose. With glucose, you can take 2 milliliter of fruit juice in a test tube, and to that we add equal amounts of Fehling solution A and Fehling solution B. Remember, it can be 5 drops of Fehling solution A or Fehling solution B and Fehling solution B, and then we boil it. For that, you need to take the test tube and hold it with a test tube holder on a spirit lamp and boil it for some few minutes. You will slowly see the color changing to orangish yellow. This confirms the test for glucose or monosaccharide. You can also take a test tube. Take a test tube, add 2 milliliter of this monosaccharide, glucon D or fruit juice. To that, add 2 milliliter of Benedict solution. and again heat it slowly. Be very careful that the test tube does not break and it's not heated for quite a long time. You will slowly see the color changing to orangish yellow, which confirms the test for glucose and monosaccharide. Next, we are going to talk about disaccharides. Take 2 milliliter of sucrose solution in a test tube, add HCl to it and gently boil it for some time. Let it cool, add NaOH to it, let it cool, and then follow the same procedure as you have followed for monosaccharides. You will get the same orangish yellow precipitate. This is a confirmed test for disaccharides. We are now going to talk about proteins. Take 2 milliliter of protein in a test tube. To that, you add a little bit of NaOH and a little bit of CuSO4 and boil it. You will find a pinkish red or violet color change in the test tube, confirming the presence of proteins. This test is called the Burette test. We can also have another test, the next one, called the Xanthoprotic test, where you can take the protein solution in a test tube and add 2-3 drops of concentrated HNO3, cool it, dilute it with water, and add a few drops of ammonia solution slowly. You will find the yellow precipitate that you have observed changes into an orange precipitate, which confirms the presence of proteins. Next test is for starch. Boil some potato, take the potato juice, add iodine solution to it, which is red in color. You will immediately see the red color changing into blue-black, confirming the test for starch. That's all.